I'm Daniel Levine, and this is the Bio Report.
While cancer immunotherapies have been promising, CAR T-cell therapies have been costly, effective on less than half of patients, and focused on hematologic cancers. Cytovia Therapeutics is enlisting natural killer cells, part of the innate immune system, to develop off-the-shelf immunotherapies to treat liquid and solid tumors. The company is leveraging a cluster of technologies to produce CAR-NK cell therapies, edited NK cell therapies to enhance their targeting, and NK engagers to redirect NK cells toward tumor targets. We spoke to Daniel Teper, co-founder and CEO of Cytovia, about the case for NK immunotherapies, Cytovia's platform technologies, and the company's pipeline of therapies in preclinical development. Daniel, thanks for joining us.
Danny, it's a pleasure to talk to you.
We're going to talk about Cytovia, its efforts to harness the innate immune system, and the company's antibody platform to develop what it calls engager antibodies to enlist NK cells in fighting cancer. Let's start with NK cells. Cytovia is not alone in looking to NK cells, but for listeners not familiar with these, can you explain what they are and the role they play in the immune system?
Sure. Now, NK cells are innate immune cells, which basically means those are the first line of defense in newborns, and obviously they remain with healthy and sick individuals over the course of life. And T cells is the adaptive immune system. So it develops as the human being matures. But I'd like maybe to put a little bit of context. The immune system has been very well known for many years, but it's really only in the last 10 years that we've targeted the immune system, including through cell therapy, to really change the way we're tackling the challenges of cancer therapy. So the first generation of immune cells that were used as treatment are T cells, and the T cells that are coming from the patient are sent to a lab to be engineered and retargeted through a CAR. CAR is like a GPS. So what we call the autologous CAR T cells are patient T cells that have been re-engineered before they're injected to the patients. And it's in the last 10 years that they came into the clinic, and they were approved in 2017. And I would say the good news is they demonstrated that in certain cases you can cure cancer, and patients that had only a couple months of life expectancy, many of them that were treated 10 years ago are still alive. So in that way, it's a true revolution. The limitations are related, first of all, to the logistics. You have to harvest the cell from the patient, send them to a lab, get them engineered, send them back to the hospital, and you lose precious time, weeks, and initially it was, even up to a month before the patient in critical situation can be treated. The second limitation, which is more associated with the T cells, not with the manufacturing and engineering, is that in a number of patients, this treatment with T cells induce cytokine release syndrome. I think many people are familiar with CRS, or cytokine release syndrome, because that's also the way, some patients are dying, following SARS-CoV-2 infection, at least in a hospital setting. You also have rejection of the cells, which is called graft versus host disease. So there's a number of issues of safety that reduce the use of those game-changing treatments to only the most sick patients, and only in the context of academic hospitals. So basically, very expensive and restricted to a small number of patients. Over the last the last 5 years, you've seen emerging allogeneic T cells and NK cells, which basically are derived from donors. So you can have cell banks, and they're available on the shelf, and they can be engineered less expensively. And then even more recently, I would say in the last 3 to 4 years, we've gone to new generations where basically immune cells are being differentiated from stem cells, from what's called iPSC, or induced pluripotent stem cells, which are undifferentiated stem cells. And through a differentiation and expansion protocol, you can make them in T cells and particularly in NK cells. And that's what Cytovia and a handful of other companies have been doing. What's very important to understand is that on the one hand, the NK cells don't have the limitations of the T cells in terms of safety, but also from a manufacturing standpoint, you can reduce the cost significantly. You basically have a monoclonal master cell bank. If you need to modify the cells through gene editing, you did it at the level of the stem cell before differentiation. So you have a very well-characterized product that then you can expand in the billions and the trillions. That reduces the cost. So what does that mean practically for patients? It means that there's a new generation of cell therapy that has only gone in clinical trials, I would say, the last 2 years, that is, remains precision medicine, is lower cost of manufacturing, and can be administered on an outpatient basis. So this is likely to expand the access to many more patients with many different types of tumors, not just in the hospital, but also on an outpatient basis.
You're developing natural killer cells derived from induced pluripotent stem cells, both unedited and edited ones, to provide improved function and persistence. What's the process for producing these cells?
So as I mentioned, there is a differentiation process, which is the secret recipe that we have and a handful of other companies have, that allows to differentiate from stem cells into NK cells. And then there's another process which allows to expand to larger number of stem cells, and then you cryopreserve them, and they're available off the shelf, and they can be used in real time by physicians.
And why do you sometimes choose to edit these? What are you editing them to do?
So you can improve the performance of the cells. You can improve the persistence. You can improve the way it handles the resistance of the tumor microenvironment, and you can do that by either knocking in or knocking out genes. And at Cytovia, we've partnered with Cellectis. Cellectis has developed the TALEN gene editing technique, and they're developing with us custom talents that allow to knock out or knock in certain genes and make the cell higher performance.
In editing these cells, do you ever edit them to target specific types of tumor cells?
Yes, and that's the insertion of the CAR, the chimeric antigen receptor is like a GPS that targets the cell to a specific tumor.
You're pursuing a number of candidates. Your lead candidates are in development for liver cancer and other solid tumors. Why start with liver cancer? What's the need and opportunity?
No, sure. That's a very good question. The first element of the response is that, many companies are technology platform companies. We're we're a therapeutic product company. So we do have those two platforms of iPSC-derived NK cells and Flex NK cell engager antibodies, but our goal is to address unmet medical needs. And liver cancer, and specifically hepatocellular carcinoma, which is the majority of liver cancer, is a huge unmet medical need. Currently, even the last generation of treatments, which is a combination of checkpoint inhibitors and Avastin, you get under 30% of partial response and only 6.8 months of progression-free survival. That's far less than satisfactory, and previous treatments had even lower response. When you look at the number of patients with HCC, it's over 700,000 patients. About half of them are in Asia, so it's very prevalent in China and in Japan. But there's also a fair number of them in the U.S. and in Europe. Probably around 40,000 patients in the U.S. and maybe 50,000 in Europe. So that's quite prevalent for cancer. So there's an unmet medical need, and we have a way to address it. And the way we address it is that we're targeting GPC3. GPC3 is expressed on HCC cells. It's also present in the circulation. So, patients that have high GPC3, and those are lower GPC3, and there's a correlation with the severity of the disease. And what we attempt to do is to redirect the NK cells toward the HCC cells, and the NK cells' role is to kill the HCC cells. So we have this approach through the NK cell engager antibodies, and this can work on its own in patients that are less severe and have enough functional NK cells. But in patients that are more severe and are deficient in functional NK cells, we have the opportunity of adding our iPSC-derived NK cells, or iNKs, as additional soldiers, because basically the NK cells are the soldiers that go and kill the tumor cells. And what we have demonstrated for now in experimental models is that the combination of Flex NK cell engagers with either peripheral blood NK cells, so endogenous NK cells, or with the iNKs, are very effective at destroying the tumor. So what we will do is we will file INDs, and then we will bring in early 2023 the Flex NK cell engager antibodies and the NK cells to the clinic. We will test each of them separately as well as in combination, and then assess what's the optimal treatment for what patient group.
You're testing your lead experimental therapy, CYT-303, both by itself and with these iNK cells. Are you doing anything to determine which patients might expect to be more responsive to one or the other?
So, CYT-303 is the GPC3 Flex NK cell engager, and CYT-100 are the iNK cells. So the first step is to do a dose escalation with the cells and with the engager, and to determine what's the optimal dose, and then go into an expansion protocol where we're combining, the optimal dose. So in the phase 1/2, the goal is to demonstrate that you have clinical activity that is better than the current treatment. And then I would say, there's a second step where you're looking to define which subgroup of patients are the most responsive. I would say that, we are in a favorable situation because GPC3 is expressed on 70 to 80% of the HCC cells, and it's not expressed at all on healthy adult cells. So there's not really a need early on in the clinical trials to differentiate patients that express it or not. I mean, if you remember back in the day, HER2, which is a marker of breast cancer, is expressed only in a minority of women. So you had to select the HER2 positive patients before initiating treatment with Herceptin. This is not the case with our treatment for liver cancer.
Is there any thoughts about testing these in combination with CAR T-cell therapies?
No, there would not be a rationale to do that. However, to test it in combination with checkpoint inhibitors such as Keytruda and Opdivo, I think would make sense. They're currently the standard of care treatment in HCC, but as I said before, even in combination, the response, the response rate is not satisfactory, and we know that patients that do not respond or stop responding to the checkpoint inhibitors have a lower number of cytotoxic NK cells. So adding NK cells should improve the percentage response.
You're waiting to begin human clinical trials. What's known about the safety and efficacy of these therapies from preclinical studies that have been done to date?
So, we know at the clinical level that NK cells are very safe, even when administered at 1 billion cells or more, multiple times and in multiple cycles. That has been demonstrated by a couple other companies. So far in the experimental model, we've shown efficacy and we've shown safety. We're still undergoing the last round of safety studies, prior to getting into patients. But we're confident, based on the initial data, that we'll pass that test and we'll be able to go into patients.
You also have a platform for developing multi-specific antibodies designed to engage natural killer cells. How does this technology work?
So I mentioned it, the 303, CYT303 is a flex NK cell engager. So it's a tetravalent antibody. It's actually tri-specific. You have, it binds simultaneously to the NK cell and to the tumor cell. In the case of 303, to the HCC cells through GPC3, and to the NK cells through an activating receptor called NKp46. It also has a fully functional FC, which supports a longer half-life, but also allows to bind to another activating receptor of NK cells called CD16A.
And how do these antibodies work to enlist NK cells in the fight against cancer?
So as I was saying just a few minutes ago, they're constructed with a flexible linker, and this flexible linker allows one arm to bind to the NK cell and the other arm to bind to the tumor cell simultaneously. So it basically by binding to the NK, activating the NK cells, it redirects the NK cell to kill the tumor cell.
Do you expect these to have value as monotherapies, or would they only be used in combination with other therapies?
We believe that, and we've demonstrated in the experimental model, that they have value in monotherapy provided the patients as sufficient soldiers, basically endogenous functional NK cells. Once that number goes down, then you want to bring additional soldiers, and that's where injecting iNK cells allows to replenish the army with healthy fighters.
I wanted to ask you about two recent transactions. You recently went public through a merger with a SPAC. Given what it's like to be a public biotechnology company these days and the availability of large funding through private capital, why did you decide to do this deal, and how much were you able to raise, and what's the case for being a still preclinical public company?
Well, I mean, it's a very good question. The majority of the companies in late preclinical stage or early clinical stage that we compete with in cell therapy are public companies. And the reason that they chose to be public companies is because the amount of capital available is much larger than for private companies. There's only a handful of private companies that get enough capital that sustain them long enough to get clinical data. So it is true that the markets in general and for biotech specifically have been challenging in the last 6 months. Essentially we had an institutional round in 2021, but by the time we were considering a crossover round and an IPO, the markets were really closed for that avenue, and we did meet the management of a SPAC that has substantial capital and trust and had a very compatible team of seasoned entrepreneurs and executives that we believed was the best vehicle for us to become a public company. So by the time of the closing of our merger, we expect to have around $100 million in capital, and that has been disclosed in the public filings and in the investor presentation. And the $100 million is 2 to 2.5 years of burn rate for the company to advance our lead programs, and particularly the HCC program.
You also recently formed Cytovia Therapeutics, a joint venture focused on developing your therapies in Greater China and beyond. What did this transaction do for Cytovia?
It's forming Cytovia was a strategic moment for Cytovia because, first of all, it brought us the first set of institutional investors. But second, it gave us access to China, which is already the second largest pharmaceutical market in the world. And I'm talking not in number of patients, I'm talking in dollar value. Also, China is the largest HCC market. I think I mentioned earlier that there was about 300,000 patients in China with HCC. So that gives us a competitive advantage in the race for new treatment in HCC, because as part of the global development, we can enroll patients not only in the US and Europe and Australia, but also in China.
What's the opportunity you see in China for these types of therapies?
It's significant given the unmet medical need and the large population. Now, I have to say that maybe many people view China as being a market that is lacking innovation. I think it has changed in the last even 5 years, including with a number of Chinese Americans returning to China. There is a lot of innovation in China, and you have companies like Legend, who is now a leader with their myeloma, CAR T therapy. So it's also access to excellent scientists. It's also, we're a shareholder of Cytolinks. So we have an interest beyond the license of our GPC3 program. We have an interest in the success of the company, and we see it as a platform, not just for the Cytovia products, but also to bring other products from the US and from Europe to the China market. And then eventually it may be a source of innovation that will also bring products either to Cytovia or to other companies in the West.
Daniel Teper, co-founder, chairman, and CEO of Cytovia. Daniel, thanks so much for your time today.
Thank you, Danny.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.